All right, guys, so in this video, we're going to be going over chapter, or at least the first part of chapter 14, which is on Mendel and the gene idea, which is what we refer to as Mendelian genetics, where we're basically making predictions about what the offspring should be. So he studied pea plants. We kind of talked about this earlier, and he chose pea plants because there's a lot of variants that he could look at. He could look at the color, he could look at the height, he could look at the color of the seed shape, inflated, constricted, green, yellow, tall, or dwarf. But you guys, believe it or not, know way more than Mendel ever could, because you know about DNA and how it's actually passed down. A lot of what he was doing was just predictions. But regardless, we can use some of his predictions to help understand how traits get passed down. So a few things about this. I'm only going to highlight some keywords here. True breeding means that they're going to be homozygous. So whenever you read that word homozygous, or true breeding, I should say, true breeding means it's homozygous. So we would have to, if you look over here to the right, purple flower, that's a true breeding purple, true breeding white. And when he crossed them, that first generation is going to always be called the F1, the first filial generation. So, and if you take a couple of purples, again, this one flower, when you crossed it with another purple flower, that's when he got this. Now, in terms of how this works out, I want you to understand that all of genetics, especially Mendelian genetics, it's a game about percentages. So you for sure need to understand that when you do this, a lot of these are just ratios and percentages furthermore. So basically when he took these flowers, about 75% of them had purple, which was the dominant trait, and about 25% had that recessive trait. Now, a few things that you want to remember too is the words phenotype. Phenotype is what you can actually see. The genotype over here is going to be the actual genes that it carries. So if you got a 3-to-1 phenotypic ratio, 3 purple to 1 white, you would get a genotypic ratio of a 1 to 2-to-1, meaning 1 or 25% would be homozygous dominant, 50% would be heterozygous, and 25% would be homozygous recessive. So don't think that your phenotype and genotype ratios are going to be the same. Now, furthermore, understand we're not always just going to be looking at traits that you can see. Your AP test will, it's more than fair for them to talk about things like if an enzyme is expressed dominant or recessive. So with that being said, let's kind of get into what alleles are. We mentioned them in previous videos, but it's just an alternative version of a gene. So basically, if you look at this chromosome, these are homologous, and basically it means that this locus, okay, and if you don't know or remember what locus means, that really means the location of the gene on a chromosome. So if we look at the locus, it's saying that these two are homologs, and at this location on both of them, it's the gene that codes for color, okay? One gene is going to essentially be the one that is expressed, the other one is not going to be expressed. Now, if it's diploid, meaning it has one of each, or I should say 2 of each type of chromosome, sometimes it could be homozygous, meaning they would be exactly the same. Sometimes they would be heterozygous. Sometimes they'd be homozygous recessive. So keep in mind, if we took a snapshot of this, like if we actually zoomed in, remember that it's all your DNA, these nucleic acids, they're the ones that are going to be getting read and getting expressed. Now, what makes something dominant and recessive is far beyond the scope of what your AP test is going to focus on, but it does help, at least in my opinion, that if you have a dominant version of a gene, basically you have enzymes that can determine that that's the non-mutated version. So if you have, let's say, white is recessive, well, that would actually be a mutation. And you have enzymes that can let the cell know that that's the faulty version, so we're only going to be expressing the dominant one. Now, if they're both mutated, the cell has to at least express it. So that's where dominant versus recessive traits come into play there. Now, and that is kind of where we're getting into. So dominant allele, it's going to determine what you would see. And then we get into recessive alleles, like I said, it's going to be masked if dominant, meaning that it's not going to be expressed. So I do want to kind of remind you guys about a couple of laws. And again, Mendel didn't know anything about these, but you want to remember law of independent assortment. It's that these alleles segregate independently during gamete formation. Okay, it basically means that if you had the Y, the S, the Y, okay, they were heterozygous for both. That when you make the gametes, you only get one of each type in meiosis after, or I should say, after meiosis. So you want to make sure that these alleles segregate independently. Okay, so again, this was random in terms of the gametes that got produced, right? So remember, if you did meiosis correctly, if you need to go back and watch the meiosis video, please do. How these gametes formed up is random. I didn't show crossing over, but remember, it's that law of independent assortment that if they lined up this way in meiosis 1, I would get these gametes. If they lined up this way, I would get these gametes. So know that how these things line up is random, but it's important to understand that. So hopefully you guys know how to do basic Punnett squares. This is a nice, easy monohybrid. So the only thing I'm going to emphasize right here is what is a test cross, because you will probably have to do this in one way, shape, or form. So when we say perform a test cross, you're going to cross with the homozygous recessive. So what is the point of a test cross? It's to determine the genotype of the dominant phenotype. So basically what this means is whenever it says perform a test cross, okay, you're always going to cross with the homozygous recessive. And why? Well, if it is expressing the recessive phenotype, you know what the genotype is going to be. Now, if let's say we had these two flowers, I don't know what this purple flower's genotype is. I don't know if this purple flower is big P or big P little p. Well, if I crossed a purple flower with a homozygous recessive, I know it's homozygous recessive because it expresses the recessive phenotype. If I crossed them and 100% of my offspring all express the dominant phenotype, well, then I know that that flower was going to be homozygous dominant. Now, if I crossed a purple flower with a white flower and I got about 50% expressing the recessive, well, then you know for sure that that flower was at least carrying the recessive allele. So that's the point of test crosses, and I wanted to put that out there. Now, when we get into monohybrid, that's pretty simple. And again, I think a lot of you guys are going to feel pretty confident on doing those. So let's just kind of emphasize dihybrid crosses. We could even do trihybrids, but that's when you have multiple alleles and you set them up and cross the gametes. So let's kind of walk you through of how to figure out the possible gametes of these. So basically, if we have this, we had purple and tall, okay? We have big P represents purple, little p represents white. I always like to put this out and remind you guys that which is dominant, which is recessive. Usually, well, they're going to typically have to tell you in some way, shape, or form what's dominant. So I'm going to say tall is dominant and dwarf is going to be recessive. Now, they might trick you and use little alleles representing dominant, so just look out for that. But let's figure out the possible gametes. Now, this is where we like to do the FOIL method, where you need to have at least one of each version of the gene. So just remind you guys, let me jump back to this one, this picture. So if you look here, if this was the diploid parent, right? Okay, big S, big Y, little y, okay? Figuring out the gametes here, it's actually pretty easy when you do this method. So let me just show you, and then we can kind of make sense of it. So if you look here at the bottom, you have one of each chromosome in your gametes, okay? So to do that, I can just make sure and do this FOIL method where one of my gametes, I'm gonna distribute this S and the Y. So one gamete is gonna be big S, big Y. I'm going to distribute that to the other one, big S, little y. And now I'm going to do the alternative one. So I have a little s, big Y. And this last one, I'm going to have a little s, little y. And you could draw the whole phase of meiosis, but it's going to give you the same thing, assuming no crossing over takes place. So big S, big Y, little s, big Y, big S, little y, little s, little y. So when it says draw the gametes, this is what you're going to be doing, okay? So when you do that, this becomes quite a bit easier. And let's look through this. Possible gametes here. If I had distributed this, big P, big T, I'm going to distribute this one. The other possible gamete here would be big T, excuse me, big P, little t. Now, we're going to run into an issue. I don't want to call it an issue, but if I distributed this again, big P, big T, and big P, little t. Notice I have some repeats. So I'm going to go ahead. If I have repeats, I'm not going to include them in the possible gametes. So I only can actually form 2 different types of gametes in this one. Okay. Now, if I had this heterozygous one, if I distributed it all, I would actually get big P, big T, big P, little t, big T, and little p, little t. Okay. I would actually get these 4 combinations of my gametes. That has to deal with how I distributed it out. Okay. So those are my 2 parents. Now, in terms of this, we could set this up, and again, I think I have it here in the slide that if this was my possible gametes of this one, okay, this is just showing you a drawing of big P, big T, little t. You could actually kind of see the relationship of meiosis here, and this is helpful, right? So you replicated it out, big P, big P, big P, big P, right? So I refer to these as sister chromatids. So after meiosis 1, they separate out, but here's my 4 gametes at the end. I got big T, big T, excuse me, big P, big T, big P, big T, big P, little t, big P, little t. Okay, so this basically shows the relationship of meiosis to what we did here for this particular genotype. Now, when you cross these, we got this figured out now. Okay, here's our gametes. Now, here's how I set up my Punnett square in this one. Remember that this is showing you this particular gametes here, that parent. I have these 4 possible gametes. These ones I set up here for this parent, right? So it's a 4x2 Punnett square because this parent can only form 2 different gametes, but this parent can actually form 4 different gametes. So that's where I have 4 and then the 2. And then to cross them, it's big P, big P, big T, big T. So this one would actually be purple and tall. This one would be purple and tall. This one would be purple and tall. This would be purple and tall. This would be purple and tall. This one would be purple and tall. So when it says what's the phenotypic ratio, I actually have 6 purple and tall. I'm going to put squares around my purple and short, or purple and dwarf. So it's a 6-to-2 ratio. Now you could easily simplify that into a 3-to-1. It'd be the same thing. Both are correct. Okay, so that's your phenotypic ratio. But when you look at your genotypic ratio, it's kind of different. It's a 1 to 2 to 1 to 1 to 2 to 1. So meaning 1 out of, what do we have, 8? 1 out of 8 would be homozygous dominant, right? 2 out of 8 would be big P, big T, little t, okay? Now, the genotypic ratio is important for you guys to understand, and I would highly advise against doing a Punnett square this way. This just shows you where this is coming from. I highly advise that you guys understand how to do the probability method. So let's kind of get this dialed in here in terms of a couple of phenotypic ratios that you should know. Anytime you see this 9 to 3 to 3 to 1 ratio, that means that you had a dihybrid cross and you crossed them with 2 heterozygous individuals. So in this case, we had big G represents green, little g is yellow, big W is wrinkled, little w is round. So like I said earlier, we did our distributive method and figured out the gametes, but notice the difference here. All of these, well, I should say both parents can make 4 gametes. So it's a 4x4 cross. Now notice this one is big G, big W. And that's important. So basically, let's talk about where this comes from. Where does this 9 come from? Well, 4x4 is going to result in 16 as your offspring, right? So if we cross these, I'm just circling all of the ones that according to these genes are going to be green and wrinkled, right? So green and wrinkled, green and wrinkled. This one's green and wrinkled, green and wrinkled, green and wrinkled. I got green and wrinkled, green and wrinkled, green and wrinkled. So basically this means 9 out of 16 are going to be green and wrinkled. Now, basically you need to catch that both of those are going to be dominant, okay? Dominant. 9 out of 16 are going to be expressing both of the dominant phenotypes. Now, what's up with the 3 and the 3 and the 1? Well, let's look at, I'll kind of do like little clouds here. Look at these ones that I'm making a little cloud around. and even the background of it kind of helps explain things. This one is green and round. Round is recessive, recessive, recessive. So it's expressing one of the dominant phenotypes and one of the recessive. So 3 out of 16 would be dominant for one, recessive for the next. Now notice that we have this one, the yellow, yellow, yellow. This one is going to be yellow, which is recessive, but still dominant for wrinkled. So the other 3 out of 16 is dominant for, or excuse me, recessive from one, dominant for the next. So, and this last one, 1 out of 16, only 1 out of 16 is going to be recessive for both traits. Okay. So that's where I want you to kind of point out that this ratio kind of helps you save some time immediately if you get these percentages. And it's a dihybrid cross, and you cross them with 2 heterozygous parents, heterozygous for both traits. So that's an important ratio to at least be able to catch. Now, let's get into the probability method, and it makes life a heck of a lot easier on you. So let's say, let's get back here. And the way that I like to do these is you set them up into their monohybrid crosses, and then it becomes a lot faster to figure these things out. So let me just erase all these. Now, let's figure this one out. And there's a couple of things that we can do here to save some time. So let's say if you have a question and you let's say it asks, what is the probability of, let's say, this particular genotype? All right. So basically, if I had a question and I crossed these two and I asked, what is the probability of getting this particular genotype? Well, you could do this and set this whole thing up. Now, it's really easy to look at now. You would just read the chart, but on your AP test, it's going to be a lot. You're going to be limited to time for sure. So what you're going to do is you're basically going to do monohybrid crosses of the parents. So let's just look at this. And then it becomes an individual, nice and easy monohybrid cross. So basically, I'm going to cross just the G's over here. So I got big G, little g. And then I have the other big G, little g. Again, where are these coming from? Basically, I'm taking these alleles. Okay, I'm just crossing the G's from the parents here. Okay. Now it would be big G here, big G, little g, big G, little g, little g, little g. And I'm going to write these proportions off to the side. So I have a 1 in 4 chance of getting homozygous dominant. I have a 50% chance, or 2 out of 4, being heterozygous. And then I have a 25% chance of being little g, little g. Okay. Now I'm going to do the W's as well. Now notice that the parents, when you cross the W's, they're heterozygous as well. So you're basically going to be getting the same results because the parents were heterozygous for those. And in this case, you can see 25% would be homozygous dominant, 50% would be heterozygous, and 25% would be homozygous recessive. Okay. Now, again, this is going to be a lot quicker to solve for this. Okay. Now, all you have to do, what's the probability of getting big G, big G? Well, you already have the proportions of these figured out. So the likelihood of getting big G is 1/4. And then you're going to multiply it by the probability of getting big W, little w. Okay. Now, the probability of getting that heterozygous for the W according to these parents is going to be 1/2. So you multiply these two together and you get an answer of 1/8. Okay. Now, that is the same thing as 2 out of 16. So I could go through and double check this. Don't do it on your actual AP exam, but notice I have this particular genotype. Big G, big W, little w. I should have another one. And yeah, I do. It's right here. Okay. And that's just one way to go back and double check, and those should be the only two. So Now keep in mind, it's quicker just to look at a table like this, but it takes a lot more time to actually build the table and analyze your results versus just doing these monohybrid crosses. So that's the probability method. I highly advise that you guys feel comfortable with that. And it goes way quicker. So let's change this one up. Let's do another one. Let's say if I had two, let's say these were the parent genotypes. If it asks for the probability of big P, big T, little t, right? These are the results of the monohybrids. You can do these on your own off to the side if you'd like, but it's the same, two heterozygous. So what's the probability of getting big P, big T, little t? Well, I'll use the 1/4 times 1/2. What's the likelihood of getting heterozygous for both? Okay, now one final thing I do have to mention would be if I added this to it, what is the probability of getting, let's say if I said big P is purple, big T is tall, little p is white, and a little t is short. Okay. Now, if I change this up and said, what is the probability of purple and tall? Okay. Well, it's not asking for a specific genotype. It's looking at a phenotype. Well, this is where we have to be a little bit careful. Okay. So if I have these two heterozygous, purple and tall, crossed with another purple and tall, they're heterozygous, and it's asking what is the probability of getting purple and tall. Let me show you this method. Well, you already established that this is the genotype for purple and tall. That's one of them. This is also purple and tall. But we also have to figure out, there's one additional genotype that could be purple and tall, big P, little p, big T, big T, okay? Now, let's figure out this ratio real quick. What is the likelihood of getting heterozygous for P? Well, it'd be 1/2, big P, little p, times, what is the likelihood of getting homozygous dominant for tall? 1/4. Big T, big T. And that would give you a value of 1/8. Okay. So basically what I'm going to do real quick is just show you how this all kind of matches up. All right. So I have a 1/8 chance of getting this genotype of purple and tall. The probability of getting heterozygous is 1/4. The probability of getting homozygous for purple and heterozygous for tall is 1/8. So if it asks the question, what is the likelihood of getting purple and tall of all of the dominant genotypes? Well, then you have to actually add all of these ratios together. And that makes it a little easier than making a big old Punnett square. So basically what I would have to do, what is the likelihood of getting purple and tall for this genotype? I'm going to basically add these all up together. It's 1/8, which I got from here, plus 1/4, plus another 1/8. Okay. Now, to do this correctly, I want to make sure that I'm not missing any particular genotype, and I am, now that I'm thinking about it. So let me go back and erase this. There's one final genotype that would give me purple and tall, and I'll do it over here. So the probability, you got to also remember you have your big P, big T, that would be purple and tall. So remember the likelihood of getting big P is 1/4. The likelihood of getting little t, big T is also 1/4. So it's 1/4 times 1/4, okay? And you have a 1 in 16th chance of getting that particular genotype that's homozygous dominant. So for this one, I'm glad I caught that because now we're gonna see where these numbers come from. So probability of getting purple and tall, I have 4 possible genotypes. I could be big P, big T, little t. I could be heterozygous. I could be homozygous dominant for tall, heterozygous, and then I can be homozygous dominant for both. So now all I'm going to do is add all these ratios up together. So this last one is the 1/16. Okay. Now, when you add all these up with common denominators, you're going to get a likelihood of 9 out of 16. okay? And that's where this is all coming into play, back to helping you guys understand and memorize those ratios. So just remember and look out for that. If it asks just for one genotype, you just have to do one calculation and you're done. But it asks for like the probability of a phenotype. That's where you have to double check and make sure, okay, well, what are all the genotypes I could get that gives me that phenotype, and then add those proportions together. So that's kind of a nice introduction on Mendelian genetics.